Hey, it's 10 after. How's your day going? Glad you asked. My day is interesting in one particular way. Right before the morning class, I shouldn't say the morning class, right before the noon class that I'm doing, my computer had the nice, unique feature that when I clicked down on anything inside of JMP, then I couldn't release it and I couldn't control things like the shut my computer down button or the activity monitor or anything like that. So effectively, I could do a few things, but not all the others. And so I was able to sort of limp my way through that class. And then in the last couple hours, what I've done is I've switched over to a different computer entirely. And so I will not promise that things will be perfect, but I do promise it'll be better than if I use the old one, the one I've been using earlier today between noon and 1. So hopefully your day is going well. Anyone want to share a thing? Well, that's not so much sharing. Okay, great. Okay, so what I've got is opportunity to maybe do a couple things. One of them is I'm going to sort of share with you a screen, the sort of thing that, hold on. Well, let's see what happens. Allow Zoom to share your screen. Fine, open system preferences. Okay, Zoom, yes, sharing, good. Oh, well, okay, so I'm going to switch gears and I have to restart Zoom, and I'll be working off my iPad while I am waiting for Zoom on my computer to restart, which means I have to assign myself to be the host. And then I need to start my audio on this machine. And that should allow. Okay, so this is an awkward situation, but you know what? I'm still here and it feels slicker than if I had done something else. But yes, I'm in the process of rebooting Zoom, so that way I can use the machine properly and share a screen with you that way. And trust me, it'll take just a matter of moments, I hope. Gosh, we'll find out. Okay, so it should be working there again, and click my join button. Okay, and I should be back. Okay, so let's just go ahead and see if we can this time share a screen. Boy, would that suck. Okay, here we go. And I'm going to go ahead and reclaim host here. And yeah, so I should be sharing a screen at this point in time. And all that was just to be able to show you other things in class today, because if I can't share a screen, that's going to really not work so well. Okay. So where do we stand? Yeah, I've got stuff for the day. You might note here that there were two week 3As last time. We're down to one of these week 3A items. And inside this week, you can spot that we had done so many explanatory variables last time, focusing on sequential sums of squares. And I know it's a fact, there will be questions on that right now, perhaps in a moment, I should say, and then also into chapter 6 and even into chapter 7. The sequential sums of squares versus adjusted is a very key topic, which is, I'll just say, it's good to ask questions about that over and over and over again if one needs to. Okay. Then we're going to be sort of moving on into experimental design today. So chapter 5, Tuesday stuff should show up there. And then the lab assigned for the day today is this design of experiments. And there's an additional set of 2 homework questions out of chapter 5 that are due on Wednesday. So administratively, do any of you have questions? And this new version of Zoom, or maybe it's the different computer, I am not seeing y'all the same way that I normally do, so I might not spot if you're raising your hand. Okay, any questions? Okay. Okay, so what I'm going to do is move this over here and sort of dive in on newish material. And I'll just say in chapter 5, we're really tackling a couple basic concepts in what I'll term, again, experimental design. Now, some of you would say, like I did at first, with experimental design, it doesn't feel like we're talking about how to design experiments, but we're talking about general concepts which, if one were to design experiments following these concepts, the analysis would be considerably more straightforward using the off-the-shelf packages that we've got that tackle ANOVA and variations on the theme of ANOVA. Okay, so let's sort of go forward and let's talk about this conceptual situation where you want to design this study that you're trying to figure out whether, okay, so let's talk grapes. When you're growing grapes, and some of you might even be wine and vit people, so you know this better than me, you have to have these vines, and the vines are in rows, and you would love to be able to treat a whole row, a whole vine row, the same way. You know, essentially sort of having, okay, so essentially, darn it. Okay, so when you're treating the whole vine row the same way, you've got this easy to work with way of tackling your whole vineyard. You know, you don't want to sort of assign an experiment in such a way that you have one individual plant receiving one treatment and the next plant up in the same row receiving a different treatment. You'd like to be able to apply the treatment to a whole row at a time. Okay, and so let's consider a question of this thing called compost, or of a cover crop, or both. And let's go with a control group of no cover crop at all, just regular whatever happens. So our treatments, let's go with black. Treatments, TTT for the treatment options, T. Okay, come on. Okay, come on. Okay, so the treatments, and I'll label them here, TTT for the treatments, are the control, okay, cover crop, and there's compost also, so control CN, cover crop CP for compost, and there's the cover crop and compost both, CP. So these are the options that we have in front of us, okay? And well, 1, 2, 3, 4 of them. And it's interesting that one of them is sort of the combination of 2 of the others. And it might be the case that compost by itself has one impact and cover crop has a different impact, but in conceptual sense, the impact of a compost and cover crop both might be different than the effect of compost plus the effect of cover crop by itself. We're going to talk about that probably a little bit tomorrow. I might call it even an interaction, and you're going to dive into an example of a situation like that on the lab today. Okay, so this is our 1, 2, 3, 4 treatments. Now let's talk about the actual physical reality of where this experiment was. It's in the Estrella area of Paso Robles, if you will, a little bit east of town. And as you're traveling along 46, maybe I'll put Highway 46 right there, you can see this ridge if you're heading out of town. A ridge is to the south, of you. So maybe I'll label that as north. And one thing we know is there's an underground river, or if you will, not really part of the aquifer, but there's water only slightly below the level of the ground, just right next to this mesa. So the mesa is again here. No, let's go with black for mesa. And the plants over at the very eastern side are quite close to this river. And grapevines can push down their root system 75 some feet. It might be easy for these plants over on the very eastern portion of the row. They might be able to easily have more access to water than the vines in the other areas. It's also fair to say that, so maybe I'll label this guy as sort of water-rich area. And then over on the very left, it gets a lot of wind. So water rich, windy. And so the rest of it is sort of this middling area. And in that middling area, it's not really clear that there's extra water available above and beyond what you put on the plants. And it's not going to be especially windy because the plants just upwind, if you will, from it are going to block the wind. In this area, the vine rows run north-south. The ones at the very far western portion, they block a lot of the wind. Okay, so let's talk about these vine rows, and let's go with green for vine rows. And within any one of these vine rows, you want to be able to treat it the same. Again, it's a real pain in the butt to say, I'm going to put compost on just the northern third of one of these rows, and then compost and a cover crop on the bottom 1/3, and a control, no compost, no compost, no cover crop. We're going to put that in the middle. Okay. We've got these 4 different treatments, and within any one of the rows, and let's go with I'll go with black again. Within any one of the rows, there's one row that you want to treat the exact same way. So all one treatment, and we've got 8 different rows here, 4 rows there, 4 rows here. And maybe of these 8, you could say of the 8, let's put, okay, undo. Let's put in yellow the left half, or the west half, and then the right half, which is again a little bit further downwind. So you could have blocks 1, block 2, block 3, and block 4. Now, I believe that entirely possible that block 4 will just have a better yield than blocks 1, 2, or 3. Block 1 might have a worse yield than blocks 2 or 3 and 4, just because of the nature of the physical reality. And when you look at any experiment, you probably can identify some factors that could possibly impact the results that are above and beyond the impact of the treatments themselves. Okay. So the natural appropriate thing to do might be something like, let's roll dice to see which of the 4 treatments goes into which of the 4 rows in each block. Okay, so let's roll dice here. The control group here was picked to be on the very left, compost, and the cover crop. That's indeed what could happen. And then when you move to the next block, again, you randomly pick who's going to be in which position. And then there's the compost or the cover crop, the compost plus the cover crop, then their control, and then finally the compost alone. And in each of these other 2 blocks, we randomly assign, because in the case of our very rightmost row, best access to water. We want each of these 4 treatments to have equal chance of having the best access to water, but all 4 of these rows are considerably better in terms of access to water than any of the other blocks. So the belief that we have these different blocks, and within a block, things should be similar-ish compared to a different block. And so we'll randomly assign treatments to potential places to apply the treatment within any of the blocks, and then we're going to be able to do our analysis quite nicely. Okay, so that's sort of a conceptual understanding of how we could go with this, and I'm going to clear this and get back into this. Okay, so really what we would want to do is to design it such that there are these different blocks, and within a block, things should be relatively uniform compared to between the blocks. And if there's a difference between the blocks at all, and we don't know that there will be, but if there is a difference between the blocks at all, it will allow us to then say some of the variability could be absorbed or soaked up by, explained by the block-to-block variability, and then randomization within there reduces that bias. And what we have is we have reducing the variability because of including the blocking information in our analysis. It's one of the other variables, like we talked about last time, that should allow us to then more clearly spot the impact of the different treatments. So here's another example, and I don't know if this is reasonable to see. Darn it. It looks like I'm showing only part of the, hold on. Okay, so are you able to see if I show you over here, or is it displaying only a portion of the screen? Could one of you unmute and tell me if you could see the entire data set over here off to the right?
We can see it.
You can? You're better than me, because on my iPad, I can't see anything over there. Okay, so good. Thank you, appreciate that. So here what we've got is a measure, I guess, in a grape called Mourvedre, which I don't know, it's a real varietal, and if you are a wine of it, you might have bumped into it at some point in time. But we measure in the grape juice the sugar content. So I'm going to call that Brix for whatever reason. That's just a way of measuring sugar content. It's one of those hydrometers, or maybe I said it wrong, but it sort of floats. And if it floats up high, it means there's more sugar content in the grape juice. If it floats down low, it's a little bit less. You're able to convert any particular altitude or height of this floaty device into the sugar content. And we believe that from cyan to cyan to cyan, from clone to clone, there may be a difference in the sugar content that we produce. Okay. So this clone variable, it's just a way of saying, I'm going to go out and I'm going to from a book of the above ground portion, the cyan, if you will, in our book where we could buy cyans, or clones, you might buy something that's genetically identical, and we're going to call it 23. That's a horrible name, but 23 and 23 and 23 and 23 and shoot, yet again 23. Each of these things called cloned 23 is genetically identical to all the other cloned 23s. Like if you take a cutting of some sort of a fruit plant and then stick it in water, then you get roots flowing off of that. Eventually, what you can do is clip it, chill it, and ship it to someone. And if someone wants your clone 23 cyan, they can order it from the catalog. Okay. Let's say it's a nominal variable. Okay. Clone 667, 115, 771, and 23. Those are the four I sort of care about comparing. And I believe that possibly one of them has a different sugar content than do the others. Okay. What about the rootstock? I'm choosing to block on rootstock because the below ground portion of the plant also may be impacting the ultimate sugar level that the plant produces. The below ground portion of the plant has something to do with how it grabs water, but there's other things as well that the below ground portion of the plant, the roots do, that may be impacting the outcome, the resulting amount of sugar. And so blocking variable, I'm going to sort of think your rootstock, because I don't really care what rootstock I use. I usually plant a rootstock that corresponds to what is going to do well in the soil conditions and in the water conditions and in the sort of the heat regimen that this plant is going to be planted in. You pick the rootstock based on where you're at, perhaps, and then the clone is based on the nature of the fruit you want to get. So does sugar content depend on which clone it is? That's my main question. But I think that maybe from rootstock to rootstock to rootstock, there may be a difference in the average sugar content that will be produced. Okay. So in the style that we had yesterday, emphasize this DOE, design of experiments, that's really not what we're focusing on, even though it looks like we're designing experiments. We're focused on the analysis in this class, and I'm going to use fit model like I had before. Okay. So the sugar content might depend on what clone or above ground portion you've got, and it may depend on the rootstock, the below ground portion, again, that you're using. I'm going to click run, and I have a lot of output. If you started using fit model, you see that these things over off to the right are produced automatically. I'm going to ignore those for the moment and zoom into the things that matter most to me right now. And I'd say I'm able to explain some 76% of the variability overall. And where are we explaining that variability? Is that due to the rootstock? Is it due to the clone? Is it due to both? Neither. The analysis of variance portion says, as its null hypothesis, neither clone nor rootstock matters at all. Let me write down a model. Here's our model. Y. And I'm going to call it Yij is equal to mu plus alpha I plus beta j. Now, just note here, the beta j that I'm referring to is not the effect of a slope or anything like that. That is the blocking variable effect. And I've got an epsilon ij. So an individual plant's level of sugar is sort of an average amount of sugar. But on top of the average amount of sugar, then there's an impact of what clone it is. Plus there's the impact of what rootstock you're using. And then around the expected amount, around the expected level of sugar, some plants are a little bit higher, others a little bit lower than that expected. Okay, so the null hypothesis in the ANOVA for the model. The whole model ANOVA is that alpha 1 equals alpha 2, and in this case I've got alpha 3 and alpha 4. Those are all the same as each other, and they're all 0. That whichever of the 4 clones we use, there is no effect on the sugar content from the clone. And beta 1 equals beta 2 equals beta 3 equals beta 4 equals, there's 5 different rootstocks. Okay, they're also with no effect either. And across these two separate portions of our null hypothesis, it boils down to saying that doesn't matter. That's just the null hypothesis, that doesn't matter. Alpha and beta are 0, whichever group you're from. Now I'm going to go ahead and remove that because that's a little bit extreme, because they might not both be 0. Our alternative hypothesis, let's write that guy down. That I guess it's not what you just looked at above. Yeah, literally, at least one of the alphas is non-zero, or one of the betas is not 0. One of these 9 separate things is non-zero in value. Okay, so I'm going to jump back to where I was. And let's jump back to where I was. In terms of JMP's output, my p-value for the ANOVA as a whole says at least one of these terms is non-zero. At least one of the alphas or one of the betas is non-zero. So in your analysis of variance for the whole model, sometimes people call it a model utility test. Is there any usefulness of any part of the model? If you reject the null hypothesis that all of the model is useless, you're naturally wondering which part of the model does have some effectiveness at predicting then the resulting level of sugar in the grape juice. Now, these are effects tests. They are just like what we talked about last time, adjusted sums of squares. So let's read this. If we want to test the hypothesis that clone matters, it's above and beyond the effect of rootstock. If rootstock itself is already in the model, does adding in clone help me? The answer seems to be yes. Our second test is, if I already had clone in the model, would adding in rootstock help me at all? And the answer seems to be, I don't see it. Let me show you this with Graph Builder. Graph Builder is, again, a way of sometimes showing the sorts of things that you'd like to be able to see. And across the clones, maybe there is a difference. But I'm going to do this separately, show you different colors for each of the rootstocks. Okay. And maybe it's a little bit hard to read, but let's bigify. And can I make this? Okay, so it's harder for me to see this without leaning in. New computer thing going on. But I would say 23 seems a little bit lower than 771. At least that seems to be true. Okay. And it does look as though perhaps the purples are a little on the lower than average side, and the reds are also a little lower than average. Okay. The green maybe a little bit above average. The blues look again on the upper end of things. So there may be across the different rootstocks a different tendency, but it's not so clear what's the case. We might be able to spot something a little bit easier if you do a connect the lines graph. I'd say this purple seems a little on the low side and the red seems on the low side. Certainly blue is at the upper end and green seems as well. Hard to read that sometimes though, so let's go back to our points. If we were to draw it a little bit differently, I could try, instead of clone being there, I'll put the rootstock along the x-axis and clone as the overlay. Here from, darn it, let me just start over. Let's have bricks here and then rootstock along the x-axis, 1, 2. It's hard to see that there's an impact of which rootstock it is on then the typical sugar content. But for clones, if I drag the clones in there and show you again who's connected to who, you'd say purple has a higher sugar content. Blue seems to have a lower sugar content, and maybe red also on the lower end. Purple seems higher than blue, at least from this picture. Okay. The insights we gain from looking at that output are what we see essentially here. And our parameter estimates could characterize things like we saw earlier, there's an effect, well, there's an intercept, the average sugar content, the effect of clone 23 or 115, 667, but 771 nowhere to be seen. Maybe what we should do is turn on under estimates, the expanded estimates. And I prefer looking at those sometimes than the parameter estimates. So the effect of 23 is a lower sugar content. 115, it's a lower sugar content. 67, about zero effect. 771, a higher sugar content. The effect of the different rootstocks, whether it's 10114 or Rapidia Gloria or Schwartzman, those are all not noticeably differing from zero. Overall, the rootstock effect in the adjusted sums of squares analysis says you don't need it. Well, let's get back into adjusted versus sequential. It's just a quick reminder from yesterday, and maybe we'll spot something. Let's turn on the sequential tests and look at them a little bit more. Hold on, there's something interesting here. Notice that the sequential tests say if clone is included in the model first, clone matters. And then if I add in rootstock, rootstock doesn't seem to matter at all above and beyond the effect of clone. But these degrees of freedom, sums of squares and F ratios, are exactly the same in the sequential as the adjusted tests. That's a curiosity. Let's try putting them in the other order just to see how that works. I'm going to recall that, and clone, I'm going to put in clone last and rootstock first before clone. Come on. Delete. Mouse down and then delete. Good. And now let's put clone back into the analysis and do the run. Again, you're going to see some output that you don't really need. So let's kill that off for the moment and get down to the things that seem to be comparable between our two separate things. And yeah, again, let's turn on the sequential estimates. So sequential tests. Okay, so let's look over here. If you put in rootstock first, it doesn't matter. But if you put in clone above and beyond rootstock, yes, it seems to matter. But I'm intrigued that if we put in rootstock first or put in rootstock second, we have exactly the same p-value. That was not the case yesterday. The reason that we have this, that the adjusted and the sequentials are the same, is we have a design experiment where we have balance in our data. Okay. Balance boils down to this. We say we have a balanced design if we have the exact same number of data points or replication for each of the different treatment combinations. There's 4 clones, 5 rootstocks. For each clone, we have one of the rootstocks. So for clone 1, there's 5 rootstocks. For clone 2, there's 5 rootstocks. For clone 3, 5 rootstocks. For clone 4, 5 different rootstocks. Those for my 1, 2, 3, 4, up through 20 separate data points. Balance gets me the ability to say, instead of saying for adjusted output, instead of saying for the adjusted sum of squares, instead of saying controlling for the effect of rootstock, clone seems to matter, I could simply say clone seems to matter, because I could say, what if clone were in the analysis first? We don't have to say controlling for the other variable. The balance is helpful in that regard. Balance literally will give us what we call orthogonality, and orthogonality implies that the adjusted and sequential sums of squares will be the same no matter what. I don't want to talk about orthogonality right just yet, but I do want to show you with our data set a way that you might be able to spot that balance. I would recommend a fit y by x. And this is only if you have 2 explanatory variables, both of which are categorical. Say maybe one of them, one of them could be viewed as the response and the other is the factor. And we're just going to look at the graphical output and find that there's exactly the same number of for rootstock Schwartzman, the same number of 23s as if the rootstock were our Gloria. In fact, we get one data point of each of these different 20 possibilities. If we look inside a contingency table, you can see ones as the actual count in each of these 20 cases. The balance graphically may be easiest to spot in the mosaic plot. Let's try something that would be a little bit different. What if, for whatever reason, and this will happen later in this term, we'll talk about this case. What about if we had this one data point was missing? Plant 19 didn't bear any fruit at all. The Schwartzman 667 case, what if that did not have any data? What if row 19 was effectively missing? If I try my, again, fit y by x case, darn it. If I try my fit y by x situation, recall what I had done, rootstock clone. Notice that we don't have this stripe with an even width across each of the different colors. For rootstock 667, there is no Schwartzman case. So here's a lack of balance. That lack of balance boils down into, well, you've got one missing data point. You don't have one of the 1, 2, 3, 4, 5, 20 possibilities. The 667 Schwartzman, there's no data there when you might have wanted data there. So if I were to use my friend, again, analyze fit model, I'm going to hit recall. Bricks 3 predicted by rootstock and clone. Now this is if we are not including that one data value. Suppose a pig came through and wiped out that plant. If I were to run that analysis, we see results that are maybe not dramatically differing, but I would spot that the sequential sums of squares, so under estimates, sequential tests, the sequential sums of squares is giving you a different result than the adjusted sums of squares would be providing to you. Okay, upshot. If you have balance, y'all good. If you don't have balance, you need to carefully consider whether sequential or adjusted is more appropriate. Let me give you one more example, and also from sort of a same experiment that this fruits student did. Okay, so undergraduate in fruit science back before we had wine and vit as a particular area. She had, again, these rootstocks and clones, the same of them, and she grew 2 plants of each variety. We only saw one of them earlier, not one of the plants, but like the average grape juice across both plants. Here's the pH of the grape juice for each individual plant. We can spot here, 2 cases here. We've got our instead of, for these first 2 observations, the rootstock clone combination is the same. For the next 2, again, the same. Next 2, the same. If I were trying to look at the balance here, I would find out if there is a balance. Well, again, clone is categorical, so I'm going to make this nominal. Clone and rootstock. Let's just see how rootstock seems to differ across. Oh, well, we've got that banding, that striping, that equal stripe. It looks sort of like a fun flag, I'd say. I didn't mean this dashy thing to be showing here. Let's turn it off. Okay. So we've got this striped flag, and that's all we care about. We have balance, and so it shouldn't matter whether we say rootstock, the effect of rootstock depending on clone, or the effect of clone depending on rootstock. Let's go ahead and verify that we don't need to make such comments anymore. Under Analyze Fit Model, we got the pH of the grapes. Well, and I'm going to put in clone first, rootstock second. Why? No reason, but because I believe clone may matter more, I'll just put it in first because I think I'll care about the p-value at the upper end first, and the lower p-value will be sort of that other blocking variable we don't care so much about. In your output, we're spotting here a couple things. One is we're able to explain 31% of the overall variability of the pH based on rootstock and or clone. The analysis of variance for the model says, I don't necessarily see either rootstock or clone is playing a role at all. However, if we dive into the effects test, well, let's just verify that under estimates, sequential produces us the exact same results as we see with our adjusted sums of squares. I would say here, if I was going with sequential, clone doesn't appear to impact then the pH, but controlling for clone rootstock does seem to impact the pH. Over here, if we have rootstock in the model already, does clone help us? I don't see it. If we had clone in the model already, does rootstock help us? Yes, it does. What it boils down to is here, there's a bit of a conundrum. We would say, does either one of clone or rootstock help? I would say, I don't yet see it. But in this test for the rootstock, controlling for the effective clone, I say I do spot that. In the model we had written down earlier, maybe there's a version of it right here. In the model that we had written down earlier, mu, there's an average pH. There's the effect of which clone it is and the effect of which rootstock it is. And the null hypothesis that can't be rejected is all the alphas and all the betas are all zeros. We can't reject that. On the other hand, in a hypothesis test that allows the alphas to be non-zero, we could say that at least one of the betas is noticeably differing from zero. The reason why we have different results from these two different style analyses is that our ANOVA for the whole model has 7 degrees of freedom. And we tackled the question overall, is anything helpful? And there's 7 separate things, which all have to be 0 according to the null. And if one of them is slightly non-zero, we'd say, I can't really see it. But within the, let's break that 7 degrees of freedom for our model into 3 degrees of freedom attached to clone variability, and 4 to rootstock variability. If we have a limitation on those 7 such that we're now saying across just these 5 groups, is there any noticeable effect? It's easier to spot it. So when we use this ANOVA tool, we can answer some questions that appear to answer another question. But we don't have exactly that other question being perfectly answered. Is any of my alphas or any of the betas non-zero? I'd say I don't see any evidence of it. Are any of the alphas non-zero controlling for the betas? Yeah, no. Are any of the betas non-zero controlling for the alphas? Oh, look, we can spot it, but barely. Because we're asking a different statistical question in our effects tests than we do in our overall model utility test, we can sometimes spot things that we otherwise might miss. Okay. This seems to be one last thing that I would want to say other than to point you towards a future class and something that you'll see in our model. I'm going to use Graph Builder for the moment and say pH. And I'm going to say maybe it depends on the rootstock. So, we see some rootstocks as having a higher average than the others. And I'm going to have an overlay for clone. I'm going to sort of connect the dots. Here's a question. Do we see something that looks pretty darn parallel across these different colors? I'm not so sure. I'm going to go ahead and fit one more model. and this is like one thing the lab asks you to do with the pig diet data. pH at maturity depends on, well, the rootstock or the clone. And now if I take rootstock here and clone here, I could cross them and have this thing that says rootstock by clone. Okay, that will allow the effect of rootstock to be different across the clones. And we could run that more complex analysis. And with that more complex analysis, we might be able to answer things a little bit differently. In the lab, you're going to be asked to, under factor profiler, look at an interaction plot. Let me show you an example of one of those before we finish out for today. So this interaction plot, let me make it even bigger so we can read it more easily. And let me smallify and smallify and smallify yet again and again, but I'm going to bigify by going down. So it's easier to now read for me. Across, we've got different rootstocks shown here in red, green, this orangish color, blue, the 3309, and a different color green for the Schwartzman. And here we see for the different clones, 23, 115, 667, 771. It looks as though this R Gloria with a 23 has a really unusually low pH. It looks as though it does seem like we have almost a set of parallel 1, 2, 3, 4 parallel situations going on here. That, yeah, literally across the 23 to 115 to 667 to 771 for all but one of those rootstocks, we see a down, up, down. But for our Gloria, we start by going up a lot and then level. So maybe our Gloria and the 23 case in some sort of synergistic way produces dramatically lower pH. The below ground portion and the above ground portion of the plant working together may, for this particular combination, have a result which is differing dramatically than the effect of the rootstock alone plus the effect of the clone alone. Parallel lines here in our interaction profile would allow us to say, yeah, we see approximate additivity as opposed to an interaction. So that seems to be about where I'd like to stop. And I noticed that I felt a little bit discombobulated with aspects of this computer not working exactly the way I wanted it to. And I also noticed that y'all didn't ask questions as we were going through. Let's just say, if you got a question, I can still answer it right now, even though you didn't ask it while we were going. Any last questions?
I had a question.
Yeah, Diana.
Is it possible for a variable to have an effect sequentially, like the rootstock after the clone, but not just rootstock alone? Did that make sense?
Let me rephrase the question, okay? If I didn't have a balanced design, then you're asking, could we have a variable which, depending on the location it appears in the sequential test, might the effect be noticeable in some particular slots, but not in others?
Sort of. A little more basic than that. What if it was just the only variable versus if it came after in sequence with another variable?
Okay, so yes, if rootstock by itself, it might not be statistically noticeable. It might not have a noticeable impact if it was the only variable in our analysis.
Mm-hmm.
On the other hand, if you add in other variables, the effect of rootstock might become clearer. Our authors talk about that with the POETS data set as an example.
Mm-hmm.
There was no trend relating the birth year with the age of death, but if you give me birth year and death year, yeah, I can predict the age of death really quite well. So sometimes, Diana, you would definitely say, I want that other variable in my analysis because it will allow me to see something that otherwise I would not see. Yeah.
Okay. Yeah, I think I was just having a hard time interpreting that if I did come upon it.
Well, yeah. And I think as you plug through the lab that is attached to today's class, some of these things will sort of jump out at you. It's like, oh, yeah, it sort of makes a difference. Perhaps the way we fit a model, like we were talking about yesterday, if you go from the front left rear, do you go along the front base and then towards the back, or towards the back and then on the back of the block? It would matter which one you put in first. Income first, there's still an effect of country type on life expectancy. But if you put in country type first, income appears to not have a real benefit to it. And income by itself, we would spot an effect, but that's not the only way it could happen. It just happened yesterday that income by itself was noticeably related to the response. But when we had country type, it was even more clear that country type played a role. So that's a situation not like the poets, but the situation you're talking about is sort of like those poets. You want to follow up on that?
Okay, yeah, no, that makes sense. And then just a quick review is that the effect tests, how is that different from sequential tests? I understand that sequential is adding it on in order. I'm just wondering how the effects are different from that. If that's okay with all of you.
Yeah, the effects test is, well, suppose I had this sequentially, but listed last. That's sometimes called an adjusted sums of squares analysis. And the p-value inside an effects test should always be interpreted. If all of those other variables were in the model, does this one prove itself as being related to the response? The sequential, well, we have, does the first one matter? Well, if the first one, whether the first one matters or not. Second line is, if the first one were already in the model, does the second one help? Does the first two, if they're already in the model, does the third one help? If the first three are in the model, does the fourth one help? Those sequential tests are only making lots of sense if they're in the right order. The effects test that you get for free is always that adjusted, and it always tackles the problem of, supposing you had everything else in the model, does this one help us? And that's usually what people do, because that's usually the question that folks have.
Okay, that
very rarely, yeah. Very rarely do people want to have a test like, well, if these three variables are in my analysis, but not those other two, does the fourth have an impact on the response? I can think of situations that are like that, but they're rare. And so usually adjusted makes more sense. So I'm glad you asked the question. Thank you.
Yeah, that clarified a lot. Thank you.
Anyone else? Okay, I'm going to stop the share and also stop the recording. And I'm going to head over to office hours and I'll be there in 6 minutes. If you want to show up then, I will be able to answer more questions in just a little bit. Bye-bye.